Thank you. Good morning. All right, I'm Sean Ainsworth, CEO of Immusoft. We are developing a cell therapy platform that harnesses the power of B cells as miniature biofactories to deliver therapeutic proteins. The specific subset of B cells that we're working with are called plasma cells. And under normal conditions, plasma cells are generating thousands of antibodies per second, pardon me. And so it's that ability of these plasma cells that we're harnessing and enabling them to produce therapeutic proteins to treat a number of different conditions. So this is a platform approach, and we've already delivered a multitude of different types of proteins, from enzymes, antibodies, structural proteins, and so we're really excited about the prospect of being able to treat a multitude of different therapeutic indications. Our initial indication is called MPS1, and we are getting ready to file our IND this quarter and look to be into the clinic next quarter, so a great time for the company. I joined in January of this year, and it's a phenomenal management team that I'm working with. So it includes the pioneers within the company that have developed this approach from the outset, and more recently we've added expertise from Retrosense Therapeutics and Juno Therapeutics, so some great success stories. And so we have a number of decided advantages over current modalities of delivering therapeutic genes or proteins. And so in the way of, if we contemplate virus delivery of a gene, systemic in particular, one of the advantages that we have is because we are autologous and non-viral, we've got the ability to redose patients. And so you can imagine that a booster shot might be applicable in instances where, for example, there's gene silencing over time, or, for example, a young child that is dosed, and then as that child grows, the protein on a per kilogram basis continues to drop off. And so we are excited about the prospect of being able to redose those patients in those instances. And then in contrast to stem cell delivered approaches, for example, we don't require myeloablation of the patients. So plasma cells, they're long-lived, they've got about a 17-year half-life, and they engraft naturally without the need for myeloablation. And looking thirdly then to enzyme replacement therapies, we expect that sustained delivery of our proteins will result in sustained reduction of substrate, which will lead to better outcomes and ameliorate or eliminate the need for these weekly infusions that are often 4 hours at a time for young children. And so plasma cells, let me take a step back, pardon me. B cells eventually do differentiate into plasma cells, and it's triggered by a B cell that recognizes an antigen. And when that happens, there's a cascade that causes the B cell to do two things. One is to replicate, and then secondly is to differentiate into plasma cells. And those plasma cells, again, those are the biofactories, and that's what ultimately we put back into the patient after they're expressing a therapeutic protein. And so from the patient's perspective, it starts at step one here, that patient is apheresed at the clinic. The product, the apheresis product, is then shipped to our cell processing center where the B cells are isolated and transposed. We use a Sleeping Beauty transposon system. Those B cells are transposed and then they're expanded and differentiated ex vivo into, there's our signal. So they're differentiated into plasma cells, and then that whole process takes about, here, let me turn off my own phone, pardon me. 3 minutes late, actually. So that whole process takes about 7 days, and then and vein to vein, it's about 10 days with the shipping and handling. And as mentioned, we've delivered several different types of proteins already. I think that really speaks to the broad applicability of our approach and the potential across, you can imagine, not just enzyme replacement therapies, but into oncology, other orphan indications, and infectious disease. So there's really a lot of upside potential here. But I like to kind of just drill in and look at one vertical, if you will, enzyme replacement therapies. We can see that there are several that are already on the market, and I think by providing an approach that eliminates the need for weekly infusions and provides a steady state protein, we've got great opportunity just in enzyme replacement therapies alone. The Sleeping Beauty transposon system, it's already known to be safe. It's being used in CAR-T approaches in human clinical studies, so that's a great indicator for us. It certainly simplifies things from a CMC perspective in terms of just the handling of the plasmid, but and then from a cost perspective, it's substantially less expensive than virus vectors and much more readily available. So the company, our approach, we think we're really benefiting from some prevailing winds in cell therapeutics, and particular everyone I'm sure is familiar with the CAR-T approaches. And so what that means is that to us, what that means is that the regulators are comfortable with these approaches. The CMC issues are constantly being improved upon and worked out, and the pharma partners are comfortable in this space. So this, I think we can leverage a lot of what's already going on in other cell therapeutics and apply that in a new space using B cells. Pardon me. And so in terms of our pipeline, MPS1 is our most advanced approach, looking to get into the clinic with that early next year. From there, we're developing a follistatin approach, which is a myostatin inhibitor that has potential application in muscular dystrophy, sarcopenia, among other indications. And then there's MPS 2 and epidermolysis bullosa programs that we have ongoing as well that we're looking to get into the clinic over the course of the next 12 to 18 months. And so in terms of MPS1, we've got some really great preclinical data. And namely, what I like about this slide, it's a little bit busy, but that horizontal bar shows the level of IDUA that we're able to get into the plasma. And those vertical bars show the level of glycosaminoglycans, which is the substrate of our enzyme. And we see that when we deliver intravenously, we get great engraftment, and that's this middle section here. And we see about 10x the level of IDUA in the plasma as compared with intraperitoneal delivery. But nonetheless, we see about the same amount of substrate reduction in both of those arms. And so what that tells us is that we're able to deliver roughly 10 times what's required to do the job in this particular animal model. And so earlier this year, we received orphan designation on our initial indication, MPS 1. We've completed our IND-enabling talks. We've got GMP manufacturing wrapping up real time. And we're looking to file the IND, get into the clinic early next year. And the STAR then calls out a point where we anticipate being able to see initial signs of biological activity. And so one of the things that I like about this initial approach is that we do anticipate being able to measure that reduction in glycosaminoglycans in these patients early on, which I think would be a great validation for our platform. So I'm working really with a great team, and I personally was a founder of RetroSense Therapeutics, which we sold to Allergan in 2016. That deal is valued at up to $555 million. Previously launched Compendia Bioscience as well. Recently, we were joined by Jason Fontenot as CSO, and Jason was head of discovery research at Juno. Cam Gallagher is our Chief Business Officer, worked with Cam at RetroSense, and fantastic CBO. Scott McIver was head of a company called Discovery Genomics, which was acquired by Immusoft in 2016, and Scott is our Chief Development Officer. And Monica Swidlitska has the background from Dendreon, taking that first cell therapeutic through to approval. In terms of our intellectual property, it started at Caltech, some IP out of David Baltimore's lab. Initially, he was developing an approach to using plasma cells for production of anti-HIV antibodies. And so Immusoft has licensed that intellectual property. We've supplemented that with Sleeping Beauty intellectual property that was out of the Discovery Genomics acquisition out of University of Minnesota initially. And we've developed a lot of in-house intellectual property know-how as well around this. And so in contemplating this meeting as a partnering meeting, I thought a slide to address that perhaps is helpful. And just by way of update on the company financing, we're actually closing on a Series B real time, and that was led by Breakout Ventures. And nonetheless, we are open to partnering discussions, and essentially, if there's interest in working with us on our platform as a way to deliver a protein of interest, we'd be interested in discussions with groups that have the need and the interest in the clinical and regulatory expertise to complement what we're doing. And I'll leave off here. We've been really excited to have some accolades in the press and awards. We were earlier this year named the most promising company, or got the most promising company award from the Precision Medicine World Conference. We were named one of the top 20 deep tech innovations by PropelX last year, and it's great to get some media coverage outside of the scientific journals as well. So it's great to see excitement around what we're doing, and I personally am very excited, and hope you are too. So thank you. All right, thank you. Oh.
I guess ERT mechanism, what kind of data do you have to show confidence that there's a kind of a cross-correction mechanism? These B cells are just naturally secreting enzyme, and it works kind of like ERT. Is that the way to think about it?
Yeah, that's very much how to think about it. And we do have animal models of MPS I. And so, yeah, the B cells secrete the enzyme. It's taken up by the mannose-6-phosphate receptor, and we've shown across a multitude of different tissue types that there's a reduction in glycosaminoglycans.
And since it's B cells only, if there's conditions where you have kind of CNS effects, would you expect to get protein production in the CNS?
So we're working on strategies to do just that. So right now, if we contemplate, the list of enzyme replacement therapies that are on the market, none of them actually do cross the blood-brain barrier. So it's recognized as an unmet need. But I think, nonetheless, being able to, again, reduce the substrate at a steady state level due to constant protein production, and ameliorate or eliminate the need to go in for those weekly or twice a month infusions is a substantial quality of life improvement.
One more?
One more. Oh, sorry.
So that CNS question just led me to think, can you adapt this technology to another cell that you can use as a delivery system that's more appropriate for being in the brain?
Another cell type, we have really focused on B cells, and it's the plasma cell kind of,
You're using their machinery.
Exactly. So the plasma cells, they're pretty much all ER and Golgi and just designed for
There
protein
isn't anything like
production.
that in the brain, unfortunately.
Yeah, not that I'm familiar with.
Okay, thanks.
Yep. So the dosing frequency, we anticipate an initial dose that will last for many years in the patients. And then at present, our expectation is that, I guess, depending on the therapeutic window, but roughly every 5 years, we would have to go in with a booster follow-on dose. All right. Thanks very much.